Assalamualaikum and hi guys, we continue our lesson. So walls surround the cell of all fungi. Walls are composite of variety components. Typically the walls contain fibrous materials bound together by sugars, protein, lipid and a variety of polysaccharides. Okay, and while for fibril materials of the wall is the largest inert, the composition of cell wall changes with time. The functional components are important in a nutrient transport, metabolisms of non-permeable substrates, communication and cell wall modifications. So this is actually quite important information that you must understand about the wall's components. Okay. Still the same thing about the cell wall of fungi. The main identifying characteristic of fungi is the makeup of their cell walls. So different type of fungi, they have their own characteristic of cell wall. Okay, so many contains of nitrogenous substance known as chitin. So chitin is actually a carbohydrate. Okay, I told you before last time about how to remember about chitin and keratin. So carbohydrate is CHO, so CH, so component of carbohydrate, so easy to remember. Chitin is a carbohydrate made from carbohydrate, so which is not found in the cell walls of plants but can be found in the outer shells of some crabs and mollusks. Fungal cell wall is metabolic point of view very different from insect exoskeletons or a plant cell walls. Okay, about the composition, I told you before, this is quite detailed composition in the cell wall. Of course, approximately about 80% of the cell wall consists of polysaccharides. Because polysaccharide is types of what? Carbohydrates. Okay, most fungi have a fibrillar structure both of chitin, okay, chitosans or zygomycotina, and beta-glucans and variety of heteropolysaccharides. The fibers are contained in a complex gel-like matrix. Protein constitute of a small fraction of wall material, rarely more than. Not all proteins have structural role. Mating, recognition, wall modification, and nutrient involved wall-bound proteins. Hydrophobins are expressed constitutively and become bound in the matrix of the walls, and hyphae emerge in 20% and often as glycoprotein. Okay, next, hydrophobins. Hydrophobins are a group of small, about 100 amino acids, 16 rich proteins that are expressed only by filamentous fungi. They are known for their ability to form a hydrophobic or water repellent coating on the surface of a subject. Okay, next, the next component is lipids. Okay, so lipids are found in wall, usually in very small concentrations. Along with hydrophobin, lipids are waxes appear to regulate movement of water. Okay, waxes is something like a candle. Okay, wax. Okay, especially in the prevention of desiccation of cells. Walls also contain a range of other minor components, including pigments and salts. Okay, next one is a pigment. You also can find pigments in the cell wall of fungus. Pigments, melanin is a variable polymer of aromatic components. The polymer is laid down in the wall after the initiation of wall formation. Melanin is important for protecting in the hyphae and spores from UV stress. Is essential for photogenesis and attachment to surface of emerging hyphae from spores. Melanin also contributes to the stabilized fractions of organic carbon in soil. Okay, again we continue. The next constituents of cell walls are synthesized in the cytoplasms, linked in the wall at the hypal tips, and polymerized and cross-linked in the cell in the wall matrix. Next, chitin and glucans are synthesized at the plasma membranes by enzymes abandoned in the membrane. So nucleotide sugar precursors are accepted from cytoplasm, linked and passed to the wall. Wall glycoprotein are synthesized in the endoplasmic reticulum, carried through the Golgi to the plasma membrane, where vesicle release the glycoprotein through the wall to the wall. Enzymes cross-linking fibril in the wall are released through the plasma membrane. So this is about chitin. I told you before, chitin is made up from carbohydrate. Okay, so chain polymer beta 1,4 and acetylglucosamine found naturally as structural polysaccharides in most invertebrates and protists. They are used as cell wall components, give strength, where each molecule contains a unit of sugar that is bonded, okay, by hydrogen bond to give it rigidity. Each microfibril of chitin gives the shape of the cell and gives strength to matured cell walls. Microfibril can be various of shapes. For example, in yeast, short and thick, and in hyperwall, long and interwoven. So this is about chitin structure. Okay, as you can see from here, septa is rich in a chitin. Microfibril of septa are arranged in the form of tangents. Septa can be strained with calcofluor white. For example, Saccharomyces cerevisiae has low amount of chitin. Okay. So again, this is about chitosan. Chitosan chain beta 1,4 glycosamine, result of continuous deacetylation of chitin, an important component in wall of zygomycete, and can be found in ascospore walls of Saccharomyces cerevisiae. Okay. So this is a structure, okay, about chitin, chitosan, and cellulose. So okay, please make sure you know how to differentiate it. Okay. Okay, we continue to the next slide later on. Thank you.